Hello friends, my name is Arun and today's video we are going to discuss about bacteria. So previously we have discussed about that microorganism. Okay, what we have seen there? There are mainly 5 types of microorganisms are present. What are that? Bacteria, fungus, algae, protozoa and virus. Okay, so bacteria is one of the microorganism, right? So let's see what is bacteria. Bacteria are microscopic living organism. Okay, what is that? Microscopic. What do you mean by microscopic? Microscopic means which cannot be seen through naked eye. It can seen only under microscope. So many of you know that our human eye without any aided glasses, okay, we can see the smallest particle that is sized 200 micrometer. Okay, the human eye with normal vision without any aided glass can see the object up to 200 micrometer. Less than any particle human eye cannot see. What is, and you know micrometer means 1 millimeter equals to 1000 micrometer or micron. Okay, so what is the bacteria size? Can you imagine? Bacteria size ranges from 1 to 5 micron or micrometer. So now you can understand, human eye can see the object up to 200 micrometer or micron. Less than 200 micrometer any sized object human eye cannot see, and bacteria size is 1 to 5 micrometer or micron. So it is not possible to see that bacteria through naked eyes. So it can be seen only under microscope. Is clear? Fine. Next, bacteria usually single-celled. Previously we have discussed about the prokaryotic and eukaryotic cells, because this cell is not developed, okay, and the nucleus is not present, so it is single-celled. Okay, that can be found everywhere. Bacteria are present everywhere in the world. There is a hot, cold, everywhere bacteria are present. Okay, some bacteria are good. What do you know? Some bacteria are good because in our human body there are lots of bacteria present. Okay, some bacteria are helpful, like probiotic. For example, Lactobacillus, Saccharomyces boulardii. This type of bacteria help us by digesting food and so many things. While others are dangerous. Dangerous means while such as when they cause infection, means when the bacteria create pathogenic effect. Okay, means cause infection. These are dangerous. So bacteria are good also and dangerous also. Fine. So any doubt about it? So now we will see that some clinically important bacteria's classification. Fine. Now we will see that classification of the bacteria as per the clinical importance. So as per the clinical importance, bacteria are classified in 3 categories. As per the shape of the bacteria, bacteria are classified again in 3 types. One is cocci, the bacteria which shape is like spherical, it is called cocci. Secondly, rod. Rod means the bacteria shape is like rod shaped or cylindrical shape, that is called rod. Third is spirochetes. Spirochetes means the bacteria shape is like spiral, that is called spirochete. Secondly, as per the gram staining, bacteria are classified again in 2 categories. One is gram positive and second one is gram negative. Okay. Thirdly, as per the oxygen demand, again the bacteria are mainly classified into 2 categories. One is aerobic and second one is anaerobic. Apart from that, there are 2 more categories are there. One is facultative aerobic. Facultative aerobic means the bacteria is mainly anaerobic but can be survived in aerobic condition. Okay. And secondly, facultative anaerobic. Facultative anaerobic means the bacteria is aerobic but can be survived in anaerobic conditions also. That is called facultative anaerobic. So these are the mainly classification of the bacteria as per the clinical importance. Okay. But majorly in gram staining procedure, this category in very vital in our clinical importance. So next slide we will see that what is the main procedure and how you distinguish the gram positive and gram negative. Okay, just for your reference only, right? Now we will see the gram staining procedure, because through this procedure we can distinguish the gram positive and gram negative bacteria. Why gram positive and gram negative bacteria is clinically more important? Because while selecting the antibiotic and coverage of the bacteria, so it is very important to know whether the bacteria is gram positive or gram negative. For that sake, we should know that gram staining. Okay, it's just a procedure. I just explain you what is that. So there are two types of bacteria. One is gram positive and gram negative. Okay, and few reagents added on both the bacteria. And how the cell of the bacteria changed, means color of the cell is changed. Okay, first we will take the reagent crystal violet. We will add both the bacteria gram positive and gram negative. Both the cell color will change in purple. Then stain color will take both the bacteria cell. So both the bacteria will see purple color under microscope. Okay. Next, if we add iodine on that, both the bacteria color will not change. Means cell color under the microscope, both the bacteria will be visible as a purple color. Okay. Next, we will add alcohol or acetone. Then what will happen? Gram positive bacteria color will remain as purple, but gram negative bacteria color will become colorless. There will be no color. Okay, that is the difference. Again, we are adding one more color, that is safranin. Okay, what will happen? Gram positive bacteria the color will remain the same, purple color, but gram negative bacteria cell color will change like pink or red. Okay, so total gram staining procedure, if the bacteria end result is purple, then that is gram positive, and if the end result is come pink or red, that is gram negative. Okay, so through this gram staining procedure, what we see, that gram positive bacteria end result will be purple. So what we can say, after the staining procedure, if the end result is coming purple, then that bacteria is gram positive, and the end result, if the end result is come pink or red, then the bacteria is gram negative. Okay, that is the difference of gram positive and gram negative bacteria through gram staining procedure. Next, we will see the structural difference of gram positive and gram negative bacteria. Okay, now we will see that structural difference of gram positive and gram negative bacteria. See the diagram. This is the gram positive bacteria and this is the gram negative bacteria. So what is the structural difference? See that blue color layer. Okay, that is called plasma membrane. This layer is present in both the bacteria. Both the bacteria having that layer, plasma membrane. Okay, so there is no difference in plasma membrane. Gram positive bacteria also have that plasma membrane, and gram negative bacteria also has that plasma membrane layer. Next layer is cell wall or peptidoglycan layer. See, in gram positive bacteria, that layer is thick layer. Okay, you can see the green color layer. That is a thick layer for gram positive bacteria. Then thick cell wall. So gram positive bacteria has a thick cell wall. Okay, or peptidoglycan. In gram negative bacteria, the layer is present, but it is thin. So gram negative bacteria, the cell wall or peptidoglycan layer is very thin. There is a difference. Okay, gram positive bacteria, that cell wall is thick, and gram negative bacteria, cell wall is thin. Next, in gram positive bacteria, there is no outer membrane. But in gram negative bacteria, there is one more layer, red color layer you can see, that is the outer membrane. So gram negative bacteria, there are three layers. One is cell membrane, cell wall, and outer membrane. But gram positive bacteria, only two layers, cell membrane and cell wall. But cell wall is thick. That is the difference in both the bacteria. Fine. So today's video we have discussed about the bacteria's classification, and we have seen gram staining procedure and we have seen the structural difference of gram positive and gram negative bacteria. Okay, thank you.